Hi everyone, welcome to this video. Today I'm interviewing Aliza Ben-Varon from Dr. Carroll's lab, and I would like you to hear more about her studies and what DRPLA work she has been up to.
Hi everyone. Thank you, Sylvia, for having me on here to chat with you. I'm really excited to share what I've been working on, and the work that I've done with Dr. Carroll has been amazing. So a little bit about me. I worked in Dr. Jeff Carroll's lab in Bellingham, Washington for a large amount of my undergraduate years. I worked with him on the Huntington's disease models that he works with. And I was really fortunate and grateful to have the opportunity to work furthermore with DRPLA. Specifically, I worked on a DRPLA mouse model. And so it was just a really cool experience that ended up being part of my master's degree.
Absolutely. You did a lot of work on DRPLA, and we are going to talk about that in a second. For everyone that doesn't know it, Dr. Jeff Carroll, he's a scientific advisor for Cure DRPLA. So tell us about what was the project goal, and give us a brief overview of the project goals as well.
Sure. So when I was brought onto the project, the main idea was we had created a mouse model of DRPLA, specifically a mouse model that imitates and specifically has a human ATN1 gene within its body. And what that means is that allows us to actually take something that we might put into a human and put that into a mouse and see what happens. And mice are really great creatures to use for this type of study because they live for a shorter amount of time than perhaps a human or a primate might. And they also allow us to see symptoms or the alleviation or the going away of symptoms after such a treatment. So in our project with this humanized DRPLA mouse model, we created the mouse model, and then we were able to test something. It's called an ASO. It's an antisense oligonucleotide. And that's a small molecule that we put into the mouse's brain, which is a very cool technique that I was able to learn. And after doing that, we were able to see how the mouse behaved over time, all the way from essentially birth until death. And so in the lab, some of the work that I really was focusing on was that drug going to work inside of the mouse in its body in the way that we expect it might? And also, what are the behaviors that might be alleviated from the DRPLA model? So for example, as we know, seizures are often present in humans with DRPLA. The mice also exhibited some of these symptoms. And one thing of interest was that drug able to change the amount of seizures or perhaps the intensity of those seizures? So that's something that the lab is able to study. As well, we're able to look at other more quantitative, or I guess equally quantitative values for the mice, such as body weight. How much do they weigh over time? As well, how are their motor skills? We combine various tests for the mice, and those tests allow us to see how do they walk? How do they behave in a cage with its cohort of mice or its group of mouse friends, we could say. And so essentially, over time, we're able to see a large amount of data that provides us sort of a schematic and a storyline for these mice on not only how do they normally interact or how do they normally behave, but also after treatment with a drug such as an ASO, what might happen and how can we help the mouse?
Mm-hmm. Absolutely. And when we developed these mice, we weren't sure if they would show symptoms that would mimic DRPLA in humans. And we were pleasantly surprised to see that they actually do. And this has become a very valuable tool that's going to be used in a lot of studies to advance DRPLA research towards the treatment. So I know the research is early on. You don't have all the results yet, and this lab is actually still working on this mouse model and testing different treatments. But can you tell us a bit more about the work that you did with this particular ASO and how this has led to further studies that are currently ongoing?
Sure, of course. So some of the really interesting findings that we were able to obtain from this study, and during specifically my part of the study, was that the mice were able, one of the major debilitating experiences in patients is changes in motor function and impairment in that realm. And so one of the things that we studied was something called balance beam. And balance beam is a test where we place a mouse onto essentially a walkway, and it's a very thin walkway. And so it begins to test their ability to balance. And we just have them walk. And a normal mouse, we call it a wild type mouse, but a mouse that does not have the disease will be able to traverse or walk across that beam very easily, within about 6 seconds or less. Whereas a mouse who has impairment in motor skills is unable to do that in that amount of time, more than 6 seconds, and even sometimes up to about a minute, which generally means that they are struggling. And you can see that as a scientist, visually when you're looking at the mouse. You might be able to see them struggling or have some weakness. And so for that type of test, what we might do is we might take animals who are untreated with any drug. They're they're either normal, such as wild type, or they're mice that have the disease, such as our DRPLA mouse model. They are debilitated or they have that impairment, and we see that difference. What we then can do is we can treat mice, both wild type and diseased mice, with a drug, with the ASO. And this small drug molecule, if it does what we might expect it to do and hopefully help the mouse, we can actually see those motor impairments decrease. We can see the mice traverse, the sick mice traverse the beam much quicker. And so what was very exciting was we saw that occur. We saw the mice that have treatment that are supposed to be sick and that are sick. They have the genetic DRPLA alteration. We actually see that they get a little bit better on this test. And it was a significant difference between the ill mice and the non-ill mice. So that was a very exciting finding. And we saw that across the board with many different tests we did. We also ran something called Rotarod. Rotarod is a test essentially of a mouse's ability to walk on a small rotating cylindrical beam. And this is again teaching testing their stamina and testing their balance. And if they fall off, their time stops. So essentially a wild type of normal mouse will be able to walk for a very long time. They have a lot of stamina, they have a lot of balance. However, a mouse that may show ataxia, that may show impairment in their motor skills, they might fall faster. And so we also saw data that the results show the mice who were treated with ASO were able to do better on that test than the ill mice that did not receive any of that therapeutic.
These are excellent results and very promising. So you, as part of the Carroll lab, is working on this DRPLA mouse model, in collaboration with a company as well. So the next steps, and you can expand a lot more on that, is to continue testing these molecules on the mice, and also to make sure that there's no toxicity associated with the treatment, to work out concentrations, also answering questions regarding when is this treatment effective, how early on do you have to treat, is it as effective if you treat once the symptoms have appeared, and so on, right?
Absolutely. Yes, these questions are really important to delve into. You know, one experiment that shows positive results or very interesting results, such as what we have done, that doesn't answer a bulk of questions that we might want to answer further down the line. It does answer many questions, of course, but a great and an important part of science is replication, is being able to see that this happens multiple times over multiple experimental, time frames. And so exactly as you were mentioning, Sylvia, it's very important to figure out if the dosing is correct, if the type of test was correct. And so as we go further into this study, which we are doing in the lab, and we are actually replicating the study almost in its entirety with a new cohort of mice, it's really important to see what are the patterns that are consistent between the two cohorts. Are the dosing values correct? One of the things that is important to know is you can't take a drug and immediately put it from mouse into human. There's a lot that is different between a mouse and a human that you need to analyze, and you need to ensure as much safety as possible. So, the question might be, well, if that therapeutic is so great, why can't we jump right into, moving into a human or moving it into a phased trial, something of that nature? And while that is very exciting news and does help to bulk the data that would help that drug go through the pipeline, it can be really challenging to just go off of one experiment. You really need to build a background for that drug. And so that's what we hope to do and what we're working towards doing with the drug that we're using.
Absolutely. And that's one of our biggest focus in terms of research and funding as well, is testing as many of these treatments as possible to work out which ones are more promising for future clinical trials. So thank you so much for explaining that to us.